Estoy que... Well, good afternoon to everyone. First, I'd like to thank to the organizers of this summer course, to Rodrigo and Andrew for having invited me to give this talk. And today I'm going to talk about the responses in cancer cells. What kind of responses? Cell migration. Because in tumor cells, there is an increased response in cell migration, which is affected by the microenvironment in which tumor cells are encountered, right? So this is called the tumor microenvironment. And the tumor microenvironment is characterized by conditions such as inflammation and low oxygen levels, amongst other factors. And this condition of low oxygen levels is called hypoxia. So what we are going to discuss now is how the tumor microenvironment, and specifically hypoxia, controls tumor cell migration. And to this end, I'm going to discuss about the novel connections between these processes and intracellular trafficking, which is via early endosomes. Okay, so we are going to be step by step reviewing these topics. So first, cell migration is an important event in all cellular organisms, as it participates in during embryonic development, in the immune responses, during wound healing, and specifically in cancer and metastasis, cell migration is deregulated. So we have a particular changes, fluctuations in the responses of cell migration. So I'm going to refer to this aspect in which cell migration is involved. Another topic to keep in mind is on the tumor microenvironment. So when we talk about solid tumors, we have an increase in the tumor mass, right, at this stage, which is followed by the dissemination of the tumor cell to other parts of the body. And here we have a particular situation that is called the tumor microenvironment, which refers to cellular and non-cellular elements that compose the tumor mass. And we have that this tumor microenvironment is changing at the different stages of tumor progression, such as in an invasive, invasive tumor cells in the local microenvironment, and at the distant metastasis. So in particular, at the tumor microenvironment, we have this condition that I talked earlier, is hypoxia, which is the decrease in the oxygen levels at in the tumor microenvironment, and the hypoxia has been associated with increased malignancy, poor prognosis, and metastasis. So when we have this condition, we have an increased incidence of dissemination of tumor cells. And this has been associated with increased cell migration and invasion, the changes in cell surface receptors, changes in intracellular signaling, and there is a connection that has been established during the last decade that is between hypoxia and deregulated endocytic trafficking. So I'm going to now define a little bit about the endocytic trafficking and the relationship with cancer. Endocytic trafficking, which is the trafficking of material within these compartments that are called endosomes, has been associated with carcinogenesis at the different stages. And as you can see, here I show a lot of reviews just to give you the impression that this is a topic very, very broadly covered by different researchers. And in this review, you can find this figure that shows that the increased, endocytic trafficking is associated with the disassembly of cell-cell adhesions, the cadherin that Andrew already mentioned, and this is part of the called epithelial to mesenchymal transition. So we have an internalization of the receptors. Also, we have the so-called signaling endosomes. So this is a very simple model of the endosome, and you have signaling coming from these endosomes to the rest of the cells, right? So this is changed also in tumor cells. So there is a lot of reviews, as I already mentioned, and I will pay particular attention to this aspect, that during the epithelial to mesenchymal transition, we have an increase in cell migration. And as you can see here in this scheme, this model of a cell that is going to migrate into this direction. And this migration requires the movement of material from one side of the cell to the other side via endosome trafficking. And this video that is available online, you can find that the cell is moving on this direction, and there is a constant assembly and disassembly of the so-called adhesions, focal adhesions with the extracellular matrix. And this is I'm going to show a very old study group that we showed in this cell. If you see in this picture, there is the formation of the leading edge, which you find in here, that is called lamellipodia, and you find accumulation of these endosomes that you observed in here at this region of the lamellipodia. So interestingly, these endosomes, these components, are enriched in a particular protein that is called Rab5. I'm going to talk a lot about Rab5, so it's better that you, if you have any questions, you can interrupt me by now with this protein. So these endosomes are accumulated at this region and are enriched in the Rab5 protein. So a little bit about intracellular trafficking. We have the plasma membrane, we have trafficking at the different endosome compartments, which are the early endosomes, the recycling endosome, late endosome, and the lysosome. And you have different Rab proteins. There are more than 60 Rab proteins in humans, and each Rab protein controls a different aspect of intracellular trafficking. And one of the most characterized Rab protein is Rab5, which controls the early endosomes. Okay, so as these are small GTPases, that means that they can be found in a GTP or GTP bound form. When you get the GTP bound form of the Rab protein, you have the active, the functionally active small GTPase. Okay, so. And I'm going to tell very briefly that this is controlled by GEP proteins, which are activators, and GAP, which are inactivators of the Rab function. I'm going to come back to this point later on my presentation. So the first thing that we made is, okay, let's measure protein, Rab5, the active state in migrating cells, right? So we performed a multi-scratch wound assay. So you have a tissue culture plate, if you have worked with that, and you make multiple scratch on that plate. And then you get a whole cell lysate, and you measure the amount of the active protein, which is by a pull down. And you can find here the form of Rab5, which is increasing during cell migration, and this is the total protein as a loading control. Okay, so we have the activation of Rab5 during cell migration. And this is a summary model, because I'm not going to talk into detail about those former studies. This is just to summarize that the early endosomes containing Rab5s in red here participate in the cell migration process by two mechanisms. First is the formation of new adhesions with the extracellular matrix and the cell protrusion, and also on the disassembly of the focal adhesions while cells are migrating. I'm going to show very quickly some information supporting this model. You can see here, this is a migrating lung carcinoma cell. Okay, so you have here the lamellipodia, which is stained for an integrin, which is a extracellular matrix receptor, integrin beta 1, Rab5, which co-localizes with the actin cytoskeleton at the lamellipodia. And here you have this nice co-localization at the leading edge, which is this region. Now, if you pay attention to this video, in green you have the early endosomes with Rab5, which accumulate at both sides, at the side that is retracting from the cell, so this side, and at the side that the cell is protruding from, providing new cellular additions. So specifically, if you see here, there's an image from a GFP tagged Rab5, which is fluorescent in green, and a marker of these focal adhesions, this adhesion with the extracellular matrix in red. And you can see a partial co-localization between both markers. And this was interesting because by watching at the video, we found that, okay, let's start. This is the adhesion in red, and the endosome in green. You see that these components co-localize, and this is followed by the disassembly of this adhesion. Okay, so this is a very time-dependent event, and you can catch at the limited window of time. Okay, and subsequently we went to observe which is the mechanism by which these compartments containing Rab5 promote the disassembly of focal adhesions. And one mechanism that was showed before was the proteolysis of the focal adhesion components, the proteolysis of the focal adhesion proteins. And this is a very, very simple and very summarized scheme of the adhesion complex, which, in which proteins such as tallin, FAC, and other components are cleaved by proteases. And we found in this subsequent study that the early endosomes, which are these compartments, are loaded with a protease which is called calpain specifically. And you can find in this 3D reconstruction the early endosome decorated with this calpain protease. And I don't show any other more details, but you can find in here that when you eliminate these early endosomes and you deplete the localization of calpain at the focal adhesions, and there is no proteolysis. Okay, this is downstream the focal adhesion. So what happened upstream in the activation of Rab5? This is a more recent study in which we show that this is the adhesion via integrins, which leads to the activation of a cascade by a kinase, which is the focal adhesion kinase, FAC, which is bound to the integrins. And this focal adhesion kinase allows to the activation of Rab5. The mechanism is by inhibiting the action of the inactivator of Rab5. So we have an inactivation of the inactivator of Rab5. The next net product is the activation of the protein. Okay, so when we have this adhesion, the cell is adhering to the extracellular matrix, we have activation of this small GTPase, which is linked to the initial stages of cell migration. Now, here I show the data by the activation of Rab5 during cell adhesion, and when you inhibit this kinase, there is no longer this extent of activation of Rab5. So with this model in mind, we went into the tumor microenvironment. So I started talking about the tumor microenvironment. So let's come back. In tumor hypoxia, there is an association with the increased metastasis and poor prognosis. And this has been associated with increased cell migration. So the question that we want is whether is tumor hypoxia related to the tumor cell migration via activation of Rab5 and the early endosomes? So to this end, we propose this model by which a tumor cell is migrating under normal conditions, but when we decrease oxygen levels by hypoxia, we have an increase in cell migration via activation of Rab5. Here's the first data in lung carcinoma cells, but we also found this in breast cancer cells and melanoma and other tumor cell models, by which hypoxia, so the hypoxia model is taking your your plates and putting inside a chamber that will is under a controlled conditions of oxygenation. This is 1% oxygen levels, and in these hypoxic conditions we have this activation of Rab5. Okay, so when we deplete Rab5 via shRNA, we found that hypoxia-induced cell invasion is abrogated when we deplete Rab5 in these cells. So cell invasion and tumor cell migration are dependent on Rab5 during hypoxia. And as I told you earlier, Rab5 associated with the focal adhesions to promote cell migration. So the next question that we wonder was on whether hypoxia this association. And this is found in this immunofluorescence, but you can find it here clearly in this co-immunoprecipitation assay, by which Rab5 is further increasing the co-immunoprecipitation with adhesion proteins. Okay, so this we propose that this is the mechanism of operating in this phenomenon. Okay, so is the activation status of Rab5 predicting the response of tumor cells towards hypoxia? So to answer this question, use the melanoma model in vitro. This is it is based on two cell lines derived from murine melanoma, B16F0 and B16F10. So B16F0 is a melanoma cell line with a low potential to develop metastasis in vivo. B16F10 is with a high metastatic potential. So and they are both isogenic. So is the main difference that. And we measure the activation of Rab5 during hypoxia, and you can find here that hypoxia didn't produce any significant activation of Rab5 in the low metastatic cell line, but it did in the cell line with a higher metastatic potential. So interestingly, we forced the system. We made an artificial system to force this phenomenon. What was that? If you observe the cell migration response in the B16F0, there is a slight increase in cell migration during hypoxia, and this is further increased in the B16F10. However, when we express the active form, the wild-type form of Rab5 in the B16F0, these cells respond with more sensitivity towards hypoxia by increasing their cell migration. On the contrary, when we express the mutant form, that is the inactive form of Rab5 in the highly metastatic cell line, there is no longer this response towards hypoxia. Finally, by using the isogenic model of melanoma cells that Andrew told you before in the previous talk, we found that by knocking down Rab5, there is a fluctuation in metastasis to the lung. And you can find here that hypoxia, when you put these cells in hypoxia, there is an increased potential of metastasis in the in vivo model. However, when we knock down Rab5, this metastasis induced by hypoxia is abrogated. So with this in mind, we have this model. There's a lot of data that I didn't show for reasons of time, the collaboration with Andrew that he already mentioned earlier, but basically we have that hypoxia promotes the activation of Rab5, and this is followed by the activation of several downstream signaling pathways which involve the small GTPase RAS, the disassembly of focal adhesions, and so on. And this is associated with increased metastasis in hypoxic conditions. However, the question that remains, I guess you are wondering, hopefully or not, which is the mechanism by which hypoxia promotes the activation of Rab5. And to this end, we went on to the adaptive mechanisms, which is via the hypoxia-inducible factor, which is a factor that is stabilized under hypoxic conditions. So HIF1 alpha, which is the hypoxia-inducible factor, is constantly degraded via hydroxylation and subsequent degradation. However, when we have the hypoxic condition, which is low oxygen levels, there is no longer hydroxylation of HIF1 alpha, and this protein is stabilized and heterodimerizes with a HIF1 beta, allowing to the expression of downstream genes. And this is the adaptive response in hypoxia. The first thing that we studied in this model is to address the dependency on HIF1 alpha. And you can find here that hypoxia promoted the stabilization of HIF1 alpha, right? And when we knock down HIF1 alpha, there is no longer stabilization in this model. So, okay, we have this model. And then we measure the activation status of Rab5. And you can find here that hypoxia allowed to the activation of Rab5. However, this activation was abrogated when we knock down HIF1 alpha. So this event is dependent on this factor. This was also confirmed by transfecting a non-degradable form of HIF1 alpha, and you can find here the activation of Rab5, and also in an alternative model, which is clear cell renal cell carcinoma, right? This model is characterized by the mutation of the VHL protein and the stabilization of HIF1 alpha independently of hypoxia. And you can find in this model that HIF1 alpha is increased, and this is associated with the activation of Rab5. So another question that I guess you are wondering, hopefully again, is that what happened with the total levels of Rab5? Total levels of Rab5 don't change. So this is intriguing because we are talking about transcriptional event, adaptive mechanism, but it's not via total Rab5. So, in fact, we observed that by inhibiting the transcription dependent on HIF1 alpha, Rab5 is no longer activated. So we are talking about activation rather than the expression. And to this end, I told you that we are going to come back to this fact, gaps or gaps. And we screened about proteins that will be depending on on hypoxia regarding their expression. And we found that several gaps are induced in hypoxic conditions. Gaps are remember that are the activation of the Rab protein. And we paid particular attention to this gap, which is alsin 2. Why alsin 2? Because alsin 2 contains putative HIF1 alpha binding elements in the promoter, and you can find here that by CHIP assay, by chromatin immunoprecipitation, we found that hypoxia promotes the binding of HIF1 alpha to the promoter of alsin 2. Most importantly, alsin 2 protein level was increased in hypoxia and in the cell model that harbors the stabilization of HIF1 alpha. And finally, we addressed whether alsin 2 was required to the events triggered by hypoxia in cell invasion and metastasis in vivo. To this end, we interfered with the expression of alsin 2 via shRNA, and we found that the decreased invasion, decreased metastasis in vivo by using the isogenic model, and the quantification is in here. So alsin 2 is required for hypoxia-induced invasion and metastasis in vivo. And this was very exciting to us. This was in collaboration with Montserrat Reyes, a pathologist at the Faculty of Dentistry. And we found that in renal carcinoma, there is this very known increase in HIF1 alpha, independently of hypoxia, but this nice increase in the expression of alsin 2. And most intriguingly, when you analyze at the tumor, non-tumor region and the tumor region, you find that there is an increase in the staining of alsin 2 in comparison with the of the non-tumor. So this is the model that we are working currently. We have hypoxia, which allows to the stabilization of HIF1 alpha. HIF1 alpha allows to the expression of alcin-2, which in turn activates Rab5, and then the downstream events in cell migration, invasion, and metastasis. To finish up on my presentation, I'd like to refer to this model. I put this on my opinion, the deregulation of Rab5 activity is not only taking place in cancer cells. We have evidence that this phenomenon occurs in early lesions, which is specifically is referred to as potentially malignant disorders. And to this end, I'm going to refer to the oral carcinogenesis in the progression towards oral cancer. You have that the normal mucosa is altered into the initial stages, which is dysplasia, the mild dysplasia, moderate dysplasia, and severe dysplasia, which is defined at the pathological pathological analysis by the extent of the alterations in the epithelia. And finally, we have oral squamous cell carcinoma. Okay, so with this brief definition, this is a summary of the findings that we observed. First, if you observe here, Rab5, we observe that Rab5 is progressively activated during the different stages of oral dysplasia. This was very interesting because it was associated with an increase in the early endosome size, which is the compartment in which it's located. You can imagine how we did obtain this data. Well, this is not like a lecture to be answering, but we can discuss this later. How was this addressed? And this was associated with an increase in the translocation of the protein beta-catenin, which Andrew defined very well on the previous presentation. So there is a nice correlation between these events. Okay, so this is here's the answer. This is not an immunocytochemistry, but it's a tissue immunofluorescence, right? So in this tissue immunofluorescence with normal oral mucosa, mild dysplasia, and severe dysplasia, we labeled at the early endosomes with a specific antigen, and is in green. And you can find that the diameter of the early endosome is progressively increased at the different stages of the oral dysplasia. This was quantified with a lot of samples, biopsies, and we found we have the quantification if you want to observe. Now, to address the activation status of Rab5, which is a biochemical measurement and all that kind of things, you cannot go into the tissue biopsies. This is very difficult. So we move to the in vitro model. This is very important if you want to complement this kind of a study. So you have tissue biopsies, but you want to measure some cause-effect experiment. So we move to in vitro cell culture model. And we have these oral keratinocytes that are non-transformed, and we have these plastic oral keratinocytes, which is a cell line established from an oral dysplasia. And we performed the pull-down assay and observed that Rab5 is augmented in the oral dysplasia cell line. And this was associated with the expression of downstream markers associated with oral dysplasia. In oral dysplasia, which is this premalignant stage, there is an increased expression of survivin, which is a protein associated with cell proliferation and survival, and cyclin D1. So these are markers of oral dysplasia. And we observed that in vitro we found these markers, but the intriguing thing is that when we express the mutant of Rab5, there is a decrease in these markers of oral dysplasia. Surprisingly, these genes, not surprisingly, we already knew that, but these markers, these genes are downstream genes of the Wnt beta-catenin pathway. Okay, so the Wnt beta-catenin pathway, as I already mentioned, is controlled by the protein beta-catenin, which is constantly degraded by the destruction complex at the cytoplasm, and when this protein is stabilized, it can go to the nucleus and allow to the transcriptions of genes surviving encyclic D1. So, what is the relationship of this and Rab5? Okay, we have previous studies from Montserrat in which beta-catenin was progressively translocated to the nucleus in oral dysplasia. So there is a correlation between the activation of Rab5, beta-catenin, and the target genes. So we performed a cause-effect experiment by using this plastic cell line, express the mutant form of Rab5, which is found here, and you can observe that only cells transfected with the mutant of Rab5, there is no longer accumulation of beta-catenin in the nucleus. Okay, so this was also complemented with fractionation, that kind of things. So this is the work in which we propose that the activation of the Wnt beta-catenin pathway in oral dysplasia is associated with the inhibition of the destruction complex of beta-catenin and the translocation of beta-catenin to the nucleus. So what is the evidence to propose this? We observed in the tumor biopsies, not tumor biopsy, in the biopsies of oral dysplasia, that this increase in the early endosome size was associated with the recruitment of these components of the destruction complex. So the destruction complex is being sequestered into the early endosomes and then the late endosomes, and beta-catenin is no longer degraded. So this is the model that we proposed. So I'd like to finish up by telling that this deregulation mechanism of Rab5 activity is not only in cancer cells, but also in early oral lesions. I'd like to thank as I said, the invitation again, Rodrigo, Andrew. I'd like to thank the funding for this project, the Faculty of Dentistry, obviously, and the team that collaborates with this, with my group. And particularly, I like the persons that have been involved in the data that I already showed. However, for Hector and Daniela, which are here, I didn't show any of their data, but you can find them on the poster, so I invite them. I invite you to check on their data. Thank you very much.
Question? Parece que tienen ya.
Sí.
Se le acabó la cosa. The dynamic proton is essential for the different pathological neurons.
Sorry again?
The dynamic of the proton, for example, is also targets for one. In this case, do you have information about this relation between the Rab5 and the of the proton in your great results?
The dynamic of the.
For example, the pH intracellular.
Oh, sorry, yeah.
The soy protein channel in tumor is the big prolactin for the genesis and the tumor progression. And the prolactin for the both process is very complicated because the two process is a core-prolactin for the pattern.
Yeah. So you mean about a potential relationship within the Rab5 activation and the pH that could be fluctuating? Yeah. I don't have any precise information about that putative relationship. However, as you asked, there is a relationship with another kind of other transport of ions such as calcium. And calcium is very relevant because this is important for the activation of the protein such as calpain in the disassembly of the focal adhesions. We intended to measure the fluctuations of calcium in these cells, but we didn't find any significant fluctuations. However, we bet that is very narrow to the early endosomes compartments, and that's why we didn't find any overall fluctuation. But we cannot exclude that possibility. Yeah.
It's
That's,
an option because the calculus model for the activation for the proton,
Mhm.
proton dynamics is
Exactly.
one. The second question is, do you have the measure of the angiogenesis target, for example, another?
Yeah, that's a very interesting question because there is a group, an Italian group, Giorgiosita, which they showed that the signaling axis stemming at the early endosomes, which is Rab5, TIM1, and RAC1, is very relevant for the angiogenic responses. So we binding of growth factor receptors, which allow to the recruitment of a GEF of Rab5, which is ring 2, and this allows to the activation of this axis. And this is followed by the increased migration of endothelial cells and then more angiogenic responses. Yes, this has been addressed elsewhere, but we don't but we don't have any evidence that this is operating in our model, and also any connection with the BGFA. That it's a nice possibility. We have evaluated that, and because BGFA is a target of HIF1 alpha and also for beta catenin. So, yeah.
Yeah, because of the both are complementary and
Exactly.
progress. It's
Yeah.
very poor pronounced in the
Exactly.
reason for the question.
Yeah, yeah. It's a very interesting possibility. We don't know yet about that.
Thank you for
Mm.
explaining.
Thank you.
Thank you very much for your presentation. I have a technical doubt because recently I saw a seminar about the hypoxic condition, normoxic condition, and many of the cellular studies are doing in 21% of oxygens, and this is hyperoxia. Actually, 5% of oxygen is what we call normoxia inside body. And hypoxia is about 0.5, but when we enter in hypoxia, which is a small box, we get out the cells of this box and obtain the cells. And when we take out the cells, we expose the cells to hyperoxia that triggers the activation of HIF1 agonist. So, how to address the problem?
Yeah, there's a lot of technical issues with that. First, maybe, I don't know if you want to take a break or no, I can tell it because it's long. Not long, but we established the conditions for the hypoxia because there's an equilibration timing on the culture media, and it's not that fast. So you need like 3-4 hours to equilibrate the gases with the culture media. So we pre-hypox the media, and then we change the media to the cells and put them in the hypoxic condition, and that's the time point 0. Okay, let's go through hypoxia, and then we remove the material. Then you have the same kinetics for working with your conditions. To minimize that, you can imagine all that kind of tricks that you can do, working on ice, stopping all processes, being fast. But when you homogenize your cells, your HIF that you can obtain is very labile. And so Daniela can tell you, or Hector, they have been fighting for measuring HIF1 alpha. You take a little bit on your tube, and then it's degraded because it's very labile. So then you have to work very fast with that kind of things. Okay, I don't know if that was we can discuss more details about that, but yeah, there's a lot of tricks in analyzing.